Hello, we are from Lyndonville High School, and for the 2019 Bio Expo, our group did Bioprinting and Neurology. In this presentation, we will cover the basics of the human brain, introduce what bioprinting is, go over the 4 main types of bioprinters, discuss the applications of bioprinting today, and provide real-life examples. We sincerely hope you enjoyed the video and learned something new. The human brain is a mysterious part of the body that makes us human. Although there is still a lot of discoveries to be made about the brain, scientists have found out so far that the brain is divided into different lobes, which are responsible for the different functions humans can execute. For example, the frontal lobe is the largest area of the cerebral cortex and is at the top front part of the brain behind the eyes. This lobe acts as the brain's central executive and is believed to be important for seeing consequences, pursuing goals, and maintaining emotional control. The parietal lobe is behind the frontal lobe but still on top of the brain. This part of the brain sends signals to muscles and controls voluntary movements. The occipital lobe is located at the very back of the brain and is furthest from the eyes. This can be anti-intuitive because one of the major functions of this lobe is to interpret messages from the eyes and the visual cortex. The final lobe that we will be discussing is the temporal lobe. The temporal lobe is where the sounds sensed by the ears is processed. For the remaining parts of the brain, the cerebrum is located towards the front of the brain and is responsible for integrating complex sensory and neural functions. Next, the cerebellum is located at the bottom rear of the brain and is responsible for coordinating some habitual muscle movements. And finally, the spinal cord extends from the brain to the rest of the body and serves 2 functions. One is to connect the peripheral nervous system to the brain, and the other is to coordinate simple reflexes. Now that we have a basic understanding of the brain, let's go talk about bioprinting. Bioprinting is the process of 3D printing viable cells to create biological matter for a medical purpose. Currently, there are 4 primary types of bioprinters, each having their own pros and cons. First of all, we will start with the inkjet bioprinter. The inkjet bioprinter has an ink viscosity of 3.5 to 12 megapascals per second, low cell density, high resolution, fast print speed, cell viability of greater than 85%, and low cost. Inkjet bioprinting has opened the way for tissue engineering. This bioprinter utilizes the technology of drop-on-demand so that printing of complex and precise sections of living tissues is possible. For the bioink, cells and biomaterials are utilized. The extrusion bioprinter has an ink viscosity up to 60 million megapascals per second, no cell density limitation, moderate resolution, slow print speed, cell viability of 40 to 80%, and medium cost. Extrusion bioprinting involves creating organized tissue by simultaneously distributing cells and matrix materials, which would repair or replace damaged or diseased tissues and organs. The laser-assisted bioprinter has an ink viscosity of 1 to 300 megapascals per second, medium cell density, high resolution, medium print speed, cell viability of greater than 95%, and high cost. The laser-assisted bioprinting gives the engineers control over the cell density and 3D tissue constructs. This gives this bioprinter a lot of potential for creating living tissues with physiological functionality. The stereolithography has no limitation to its ink viscosity and cell density. It has a high resolution, fast print speed, a cell viability of greater than 85%, and a low cost. Recently, there has been greater improvements in the quality, resolution, and accuracy of stereolithography. This technique, along with medical imaging technology, is helping to improve the diagnosis of disease, preoperative planning, quality and morphology of prosthetics, implants, and complex surgery. The stereolithography has been the primary tool to advance regenerative medicine for the future. According to research conducted by the National Center for Biotechnology Information, 3D printing has been proven to be a practical and anatomically accurate method of producing patient-specific models for surgical planning, simulation and training, tissue-engineered implants, and secondary devices. Therefore, the conclusion is that advancements in bioprinting would also translate to advancements in the field of neuroscience. Because most diseases neurosurgeons face are intricate and detailed, having the physical models available to them helps the surgeons to visualize the problem better compared to the images produced by X-rays, CT scans, and MRI scans. Additionally, having the physical model of the brain enables neurosurgeons to have a surgical simulation which allows the surgeons to practice surgery without the potential risk of harming the patient. Likewise, the models of the brain have also been used as prototypes by manufacturing industries to run test trials of their instruments. Overall, the anatomical models of the brain with bioprinting allows for the improvement of surgical planning, training, and education, the design of neurosurgical devices for assessment and treatment of neurosurgical diseases, and the development of biological tissue-engineered implants. One real-life example is the story of Bon Verwage, who is a neurosurgeon at the University Medical Center at Utrecht. In 2014, Verwage used a bioprinted brain to replace a patient's entire skull. The patient had a rare condition where the skull will constantly grow and pressure will be put on the brain. But now, the pressure on the brain is gone and the patient has a 3D printed skull. Also, in some hospitals, the application of using 3D printed brains for practice surgery has been revolutionary. For example, some hospitals in Ohio and in San Diego have implemented this new method as a way to better treat their patients. Also, at Harborview Medical Center, 3D printed brains are being used to help medical students. In Washington University, they have also used this method for hearts instead. Bioprinting is a growing field that has a large influence on modern medicine. There are many applications of bioprinting in neuroscience, and new developments in this field help us to understand our brain better. The tool of bioprinting has greatly refined and advanced our practices in neurosurgery, and it has also been used to print neural tissues for cell therapy. In conclusion, bioprinting remains an emerging and growing technology that has incredible potential in the field of neuroscience.